In this lecture, we will study time-resolved fluorescence and determination of excited states and lifetime. Fluorescence is always an issue of excitement for researchers. Fluorescence studies can give lot of information about the samples, and especially when we study time resolved, how how sample behave with respect to time. Initial study can be obtained without time resolved study, but if you wish to go into detail of the samples, then this study is very important. We have discussed vibrating interactions, Heisenberg-Taylor theory, then fluorescence spectroscopy, Kasha's rule, quantum yield, non-additive transitions, Jablonski diagram, and this is the topic to determine excited states and lifetime. We will discuss in this topic. In this lecture, we will study basics of time-resolved fluorescence, the dependence of various parameters like concentration, emitted uh, excited spectra, then temperature, then intensity, concentration, and we will discuss a brief about lifetime and intensity measurement, which is better, how it is better than steady state, time resolved is better than steady state, and then we will discuss some important example of new techniques. The time resolved fluorescence and determination of excited state. The time resolved, how the time factor come into picture in fluorescence, and how the studies can be carried out to get the ideas of excited state we will discuss. We have discussed in the previous lectures, and we all know that the emission spectra for a molecule, when we excite a molecule by some excitation, has multi-parameter nature. Multi-parameter nature means the behavior depends on many parameters: time, wavelength, intensity, what intensity you are using to strike, and what is the concentration, what is the temperature, and so many things. The objective of the measurement is therefore to gain information concerning as many parameters as possible. Many times I suggest students, even you can, it's just such a vast topic that you can do almost a doctoral work on any sample if you just want to study the emission spectra. A lot of information can be obtained. It depends on what facilities you are having, mathematical facilities, experimental facilities, and what knowledge you have. A steady state measurement of the fluorescence emission. We have studied intensity versus wavelength. These are the normal graph. Gives an average and also relative representation. The fluorescence lifetime give an absolute measure and allow a dynamic picture of fluorescence. Factors that explain the appeal of the form of presence measurement. We know that the decay of excited state of a molecule to the ground state is given by this expression: IT is equal to I0 exponential of minus t over tau. This I0 is the intensity at time 0. Time 0 means initial. Here you can use the word, this is wrong word, use this I0. This is the maximum intensity. Here tau gives the idea of your lifetime. Zero. Lifetime means whenever you give the energy, it moves to excited state, and at what time, up to what time it can live there. This is defined as the time of the intensity to drop 1 by E or 37%. This is called lifetime. In terms of rate constant, radiative rate or non-radiative rate, we have Kr or Knr, we use two words. Generally, either the transition is radiative or non-radiative. So we have two types of radiations. And in terms of this, the lifetime can be done as below. The lifetime can be done as tau is equal to 1 upon Kr plus Knr, or phi is equal to Kr upon Kr plus Knr. The fluorescence signal is multi-parametric as we discussed earlier, and can be considered as follows along with the measurement that can illustrate them. Since it is multi-parametric, I am writing, suppose it's a function of many things. So we are writing the equation as Fl is equal to function of many things, where I is the intensity, lambda excitation wavelength, emission wavelength, then polarization, measurement of anisotropy, then measurement of fluorescent microscopy, X, then time of fluorescent time. So we have clubbed all these parameters on which it depends. What is the emission wavelength, what is the excitation wavelength, what is the polarization, what is the position of the microscopy. So we have put at the one end. Although the decay law is based on first order kinetics, in practice, many fluorescence decays are more complex. Often, population of excited molecules are in inhomogeneous environment, and quenching process and other environment influence can lead to multi or exponential decay behavior. So we have to take care of all these things. This system is inhomogeneous, not homogeneous. Quenching process is there. Many environmental influences are there, so it complex the system. One of the major advantages of using fluorescence lifetime is the fact that it is an absolute measurement. Lifetime is an absolute measurement, unlike the steady state intensity, which is relative. For somewhere the intensity is different, and sometimes the intensity is different. It's a relative about the measurements, but lifetime is not relative. The fluorescent lifetime is an intrinsic molecular property, and within certain constraints independent of concentration, whereas your intensity may depends on concentration. This means that the changes in concentration, whether caused by photobleaching or diluting concentration, the sample would not affect the lifetime. This is in contrast to steady state measurement, where a change in intensity recorded would be observed. That is why lifetime measurement is very essential or beneficial than other measurements that we do. In a single photon counting, that's called TCSPS, time correlated single photon count, counting, the lifetime is not influenced by fluctuation of source intensity. This implication is that a measurement can taken on one piece of equipment at a certain time and place should be capable of being repeated later. Normally, this is the biggest challenge with the fluorescence. Whenever we take the calculations, we always have such type of issues, intensity issues, repeatability issues. So, if you are working with lifetime, lifetime is fixed. Lifetime it depends on certain things that it is fixed for the material, and that is why lifetime should be used. It may be asked why we choose lifetime, why time is always important. That is why it is important. These information cannot be easily repeatable with only conventional wavelength intensity curves. Fluorescence is an ideal nanoscale probe as the fluorescence decay takes place on nanosecond time scale and it can be influenced by molecular process occurring on the nanometer range. The emission of fluorophore can be highly influenced by the environment or the presence of other interacting molecules which can affect KNR. We have discussed KNR radiations, non-radiative radiations. Thus, the fluorescence lifetime is useful in elucidating tau changes in the environment. Obviously, viscosity, pH, polarity, solvation size, and the shape of molecules, interaction, inter and intramolecular distance, kinetic dynamics, and resolution of this. If we know lifetime, so we can get many things. How the viscosity is changing, how the pH is changing, solvation size, shape of the molecule, interaction, many things can be obtained with this. The extra specificity of the fluorescence lifetime allow easy discrimination against scattered excitation and background fluorescence. Determination of Foster resonance energy, that is called FRET, is much simpler using fluorescence lifetime as are quenching and fluorescent anisotropy measurement. This allowing more parameters to be recovered. Now you may be aware of this fluorescence resonance energy transfer. We are using this method in developing many systems where we transfer the energy from one system to another using this process. Energy can be transferred and we use this property in many pharmaceutical systems. We can transfer the energy and study the quenching and anisotropy measurements. However, it should be remembered that the use of steady state and time resolved techniques are complementary in elucidating as much of the multi-parametric fluorescence signature as possible. So both are required if you want to work with a preliminary stage. You can use steady state. Otherwise, time resolved is useful more. Another term we also use, that is time correlated single photon counting, TCSPC. This is considered most sensitive method for determining fluorescence time. It is a digital technique with well-defined statistics, Poisson statistics, which is not affected by changing in source intensity, unlike some analogs measurement techniques. TCSPC is based on detection of arrival time of individual photons after optical excitation of a sample. It make use of a pulse excitation source as shown in the figure. Typically, generally we use laser or LED. Production of a light starts the timing electronics. These are stopped by a single signal from detector triggered when a fluorescence photon hits the detector. This difference in the time between these two signals is then output as a histogram consisting of time bins with fixed time bits. So, signal arriving within a time go into one such specific bin. The width of the bin influence the time resolution of the system. By this we determine fluorescence lifetime. As they are shown, this is a digital technique and in which we study detection of arrival time of individual photon. First we excite a sample. Sample is there, it should be excited. Then we study the arrival time. If we found some difference between these two signals, suppose two signals coming, then it will give you a histogram. And this gives many information. The excitation emission process is repeated many times. Figure you are seeing that in this figure we have source, we have sample, and sample is giving energy. And we are analyzing the system. Histogram is built for number of events. However, for histogram to be representative of the decay, that is the number of counts at a certain time after excitation to be equivalent to intensity at that time. A certain conditions needs to be observed. This is that only one photon can be counted at any one time. So it is required to statistically count only one photon and it not arrive during the dead time of timing electronics. In practical term, the stop count rate needs to be limited to 2% of excitation rate only. Failure to do so can lead to biasing towards detection of photons. When measuring long-lived decays, re-excitation of the sample before it was fully decayed should be avoided. Decay is important. We have to be decayed because the long-lived decays are there. Thus, the excitation rate should be set to be less than 1 upon time range. Time range are normally selected to 10 to 20 times that of lifetime to be measured. Try to understand this. It's a practical issue. First, should check that lifetime. Then a time range should be 10 to 20 more than this. And for this time range, we can set excitation rate. Everything should be synchronized. This restriction on the maximum excitation rate and hence data collection rate means the measurement of long-lived decays can be quite time consuming or time to amplitude converter based system. Normally, a different approach is used when measuring decays on the phosphorescence time scale to overcome this restriction and reduce the data collection time. We have discussed time resolve. Now we will discuss some common form of fluorescence measurement, briefly summarized here. Actually, this is a practical topic, so I suggest personally develop your samples and analyze in the lab using the methods. Try to get information which sample can be analyzed using this, so it will be more useful. It will be helpful to learn the things. First one is fluorescence anisotropy. Basically, this is a measure of how ordered a molecular system is and follow its disorder via molecular rotation or diffusion with time. It used polarized light to excite a sample, and the parallel and the perpendicular polarization are monitored. It is very interesting how it is used to study the orderedness of the system. It gives the idea of the system, how the system is ordered, and so we use polarized light and we monitor the system. Another application is Foster resonance energy transfer, FRET. This is basically a Coulombic dipole-dipole interaction. This requires spectral overlap between donor emission and acceptor absorption. It is manifest all 10 to 1000 range, which makes it suited to follow molecular interaction and determine distance on the nanometer scale. If only the lifetime of unquenched donor is available, then this can be relative and made absolute if the distance at which FRET is 50% of known as system under study. Here we study allowing this way study the energy can be transferred from one system to another. So one is quenched donor, unquenched donor. It is only the lifetime of unquenched donor is available. Another is time resolved emission spectra or decay. This is useful if there is several emitting species present with overlapping emission spectra. Fluorescence and lifetime are measured at wavelength for same data collection time. The resulting intensity wavelength time surface can then simply be time sliced in intensity time axis to provide spectrum at different time after excitation. We study, actually we uh, divide here time, and we note down the after excitation we note down the results intensity after certain time interval. Alternatively, the dataset can be analyzed globally, linking common lifetime. The pre-exponential factor then relate to the amount of each species present in certain wavelength. Weighing these by their associated lifetime and plotting versus wavelength can provide spectra associated to that, called decay associated spectra. The summation of individual spectra plotted is used, and this should return the equivalent of steady state spectrum if we add all this. Thanks. In this lecture, we have studied what is time resolved spectroscience, why it is important, what is the significance, and how many types of studies can be carried out here. Thank you.